Have you ever wondered how the cholinergic system and drugs work in our body? The cholinergic system, a crucial part of our nervous system, is responsible for an array of functions. It plays a key role in memory, attention, and learning, as well as muscle movement. The star of this system is a neurotransmitter called acetylcholine. From a small molecule, it wields enormous influence, facilitating communication between our nerves and muscles, and within the brain itself. When acetylcholine levels are balanced, the mind and body hum along, but when they aren't, we can see a range of disorders from memory loss to muscle weakness. So, understanding acetylcholine and the cholinergic system is not just about satisfying scientific curiosity, it's about coming to grips with how our bodies function at a fundamental level, and how we might treat and prevent a host of conditions. Ready to dive into the world of acetylcholine? Stay tuned. The journey of acetylcholine begins with its creation. Like any good story, acetylcholine's tale starts with its birth, and it's an intricate process, to say the least. You see, acetylcholine doesn't just appear out of thin air. It's created through a complex series of biochemical reactions that take place in our body's nerve cells. To kick things off, we start with two key ingredients, choline and acetyl-CoA. Choline is a crucial nutrient that we get from our diet, found in foods like eggs, beef liver, and soybeans. Acetyl-CoA, on the other hand, is a coenzyme derived from carbohydrates, fats, and proteins in our diet. These two components meet up in the cytoplasm of a nerve cell, where the magic happens. Enter stage right, an enzyme known as choline acetyltransferase. This enzyme acts as a sort of biochemical matchmaker, bringing choline and acetyl-CoA together to form a new compound. And voila, acetylcholine is born. But the role of choline acetyltransferase doesn't end there. This enzyme is the gatekeeper of acetylcholine production. It regulates how much acetylcholine is produced based on the body's needs. When more acetylcholine is needed, the enzyme steps up its game, facilitating more reactions between choline and acetyl-CoA. And when less is needed, you guessed it, choline acetyltransferase slows things down, reducing the production of acetylcholine. So in essence, the biosynthesis of acetylcholine is a carefully regulated process closely overseen by the diligent enzyme choline acetyltransferase. It's a fantastic display of the body's ability to maintain balance and ensure that everything runs smoothly. But the story of acetylcholine doesn't end with its creation. Far from it. Once it's formed, acetylcholine has a vital role to play in sending messages between nerve cells. But that's a tale for another scene. Now that we've created acetylcholine, where does it go? Let's find out. Acetylcholine doesn't just float around aimlessly. After being synthesized, it's carefully stored in specialized sacs called vesicles within the nerve endings, waiting for the right moment to leap into action. These vesicles are like the green room for acetylcholine, keeping it primed and ready for the spotlight. The storage of acetylcholine in vesicles isn't just about keeping it safe and isolated. It's more than that. The vesicles are a crucial part of the acetylcholine release process. They're the gatekeepers, the bouncers at the door of the synaptic cleft, the tiny gap between nerve cells where the magic of neurotransmission happens. So, how does acetylcholine get from these vesicles to the synaptic cleft? It's all down to an elegant dance of cellular signaling and a very special guest star, calcium. When a nerve impulse, or action potential, reaches the nerve ending, it triggers the opening of voltage-gated calcium channels. This allows calcium ions to flood into the nerve ending. Now, calcium might be a small ion, but it's got a big job. It acts as a key, unlocking the vesicles and allowing them to fuse with the nerve cell membrane. This fusion creates a pathway for acetylcholine to spill out into the synaptic cleft, ready to bind to its receptors on the next nerve cell. This process is known as exocytosis. It's like the curtain lifting at a theater, revealing acetylcholine to the audience of receptors waiting in anticipation. But this isn't a one-time show. The calcium channels close, the vesicles are reformed, and the process can start all over again. The cycle ensures that the nerve cell is always ready to send its chemical messages whenever they're needed. Once released, acetylcholine has a crucial job to do. It's the star of the show, ready to perform its role in transmitting signals across the synaptic cleft, but that, dear listeners, is a story for another scene. Scene script. Acetylcholine is now ready to play its part. With the stage set, acetylcholine, our chemical protagonist, steps into the spotlight, ready to perform its role in the grand spectacle of body function. It's taken up by nerve endings and released into the synapse, the junction between two nerve cells. Now it's time for the magic to happen. Acetylcholine, like a key fitting into a lock, binds to cholinergic receptors located on the surface of the next nerve cell. These receptors are special proteins that act as gatekeepers, only allowing specific molecules to bind. Picture it as a grand ballroom dance, where acetylcholine is the dancer and the cholinergic receptor is the dance floor. The dance begins when acetylcholine finds its partner, the receptor, and they start to move together in harmony. This dance triggers a response. In the heart, it might slow the pulse. In the gut, it could stimulate digestion. In the muscles, it might cause contraction. Acetylcholine's dance can set off a cascade of reactions, creating a symphony of physiological responses that maintain our body's delicate balance. But like all good things, this dance must come to an end. Enter acetylcholinesterase, the enzyme bouncer of our metaphorical ballroom. It breaks down acetylcholine, essentially stopping the dance. The products of this breakdown, acetate and choline, are then whisked away back into the nerve cell for recycling. This breakdown is crucial. It ensures that the response triggered by acetylcholine doesn't continue indefinitely. It's like the curtain falling at the end of a play, signaling that the performance is over and it's time for the actors to leave the stage. And just like that, acetylcholine's journey is complete. But what happens when things don't go as planned? Indeed, when the delicate balance of this system is disrupted, it could lead to various disorders. But that's a conversation for the next scene. Stay tuned. Sometimes the cholinergic system needs a little help. When it does, we turn to cholinergic drugs. These are medications that either mimic the action of acetylcholine or inhibit its breakdown, effectively boosting the cholinergic system's function. Two major categories of these drugs are parasympathomimetics and anticholinesterases. Parasympathomimetics, also known as cholinergic agonists, work by directly stimulating the cholinergic receptors. They essentially mimic the action of acetylcholine, hence the term mimetic. On the other hand, anticholinesterases obstruct the enzyme acetylcholinesterase, which is responsible for breaking down acetylcholine in the synaptic cleft. By inhibiting this enzyme, anticholinesterases allow acetylcholine to remain active for a longer period, thereby enhancing its effects. These drugs play crucial roles in managing several disorders. Take glaucoma, for instance. This eye condition is characterized by increased pressure within the eye, which can cause vision loss. Parasympathomimetics, such as pilocarpine, are used to decrease this intraocular pressure. They work by constricting the pupil and opening up the drainage angle of the eye, which facilitates the outflow of the aqueous humor, the fluid responsible for the increased pressure. Then there's Myasthenia Gravis, a neuromuscular disorder that causes weakness in the skeletal muscles. For patients with this condition, anticholinesterases like pyridostigmine are a godsend. By preventing the breakdown of acetylcholine, these drugs increase the availability of this neurotransmitter at the neuromuscular junction, enhancing muscle contraction and strength. Of course, these drugs are not without their side effects. Overstimulation of the cholinergic system can lead to symptoms such as excessive salivation, abdominal cramps, and even slow heart rate. But with careful dosing and monitoring, these side effects can be managed. These drugs show the power of pharmacology in treating cholinergic disorders. They highlight the importance of understanding not just the disease itself, but also the underlying biochemical and physiological processes that can be targeted for therapeutic intervention. Let's recap what we've learned about the cholinergic system and acetylcholine. We embarked on this intellectual journey by understanding the cholinergic system and its critical role in our bodies. We then dived into the fascinating process of acetylcholine's biosynthesis, the intricate dance of chemicals that leads to its creation. Next, we explored how this neurotransmitter is stored and released, a delicate balance that ensures our bodies function correctly. We also delved into acetylcholine's action, its metabolism, and how it's broken down after performing its vital role. Finally, we examined cholinergic drugs, those medical marvels that mimic or block acetylcholine's action to treat various disorders. We discussed how these drugs are used to manage conditions like glaucoma and myasthenia gravis, and even to treat organophosphorus poisoning. And that's a wrap on the cholinergic system and its drugs. Subscribe for more because you won't find me later.